Alright, so in the interface, we have it made up of the GS and G2 phases. The G1 is growth and normal metabolic roles, where cellular contexts excluding the chromosomes are duplicated, and then it's the S, where DNA replication is taking place, and each of the 46 chromosomes is duplicated by the cell. And then lastly, we have G2, growth and preparation for mitosis, where the cell double checks the duplicated chromosomes for error, making any needed repairs. Okay, now we have the meiotic phase, consisted of the prophase, metaphase, anaphase, and telophase. The prophase is where chromosomes spiralize. And then we have metaphase, where chromosomes align with centromeres on the equator. And then we have anaphase, where they separate into daughter chromosomes and move to opposite sides. And then we have the telophase, where two nuclei reform at poles.
Okay, so mitosis is the part of the cell where the replicated chromosomes are separated into two new nuclei. Then that is made up of the prophase, prometaphase, metaphase, anaphase, and telophase. Meiosis is the cell division that reduces chromosome numbers by half. So that means it creates 4 haploid cells, and all the cells are different from their parent cells.
Alright, up next we have cytokinesis. Cytokinesis is the part of the cell division in which the cytoplasm of the eukaryotic cell is divides into 2 daughter cells. It also is when the spindle apparatus partitions and transports the duplicated chromatids in the cytoplasm of the 2 daughter cells. More or less, cytokinesis is when the cell's already in its last stage and it's already ripping apart. It's already becoming, except for one cell, into 2, and this is the duplication phase per se. Alright, up next is checkpoints. Checkpoints, there's multiple checkpoints in the eukaryotic cell cycle, but here we're going to mention the 3 most important ones, which is G1, G2, and the spindle checkpoint. A checkpoint is technically a stage in the eukaryotic cell cycle where it measures its internal and external structures of the cell and sees if it's compatible enough to divide. In the G1 checkpoint, it's the main decision point of cell, and it chooses whether to divide or not. And it usually checks for the cell size, the nutrients, growth factors, and DNA damage. And G2 checkpoint, which is right after G1, it will check for the DNA integrity and DNA replication. And in the spindle checkpoint, it will examine whether all the sister chromatids are correctly attached, and it will check for chromosomes attached to the spindle at metaphase plate. Our last one is crossing over, and it occurs during prophase when homologous chromosomes overlap each other and exchange genetic material, causing recombination, and it usually only happens with the X